دكتور اسماعيل.
دكتور اسماعيل.
Hello everyone.
ربنا
I have problem today with my laptop to introduce to the lecture, so I will try to do it from the phone. Inshallah, it will.
Our next presentation will be
Inshallah.
Dr. Ismail. Dr. Mujahid, you can present Dr. Ismail and his lecture.
Embryology on development of the brain by Dr. Ismail. Regarding embryology and development of brain, yeah?
Yes.
Dr. Ismail is a consultant neurosurgeon, is assistant prof in 21 September University. And he will present his lecture now. Are you ready, Dr. Ismail?
Just a minute.
I think you didn't share the presentation, you shared the. Let's see. Yeah. Oh, yes, I think now it's
okay
good.
now?
Now I see it, yes, it's good.
It's okay.
Now it's okay now.
Yeah,
Okay.
it's okay. Start, please. It's okay.
Sorry for this problem. So I will talk about the embryological development of the central nervous system. First of all, the first stage of this development is usually by the junction of the sperm and the ovum to form a fertilized oocyte. After that, this oocyte, the mature oocyte or the fertilized oocyte, is divided to two cells. The two cells undergo multiple division to form about 16 cells, which is called the morella. This morella undergo more differentiation to form a fluid inside this group of cells. It's called the blastocyst. So the blastocyst consists of three components. The first one is the fluid and the two groups of cells. This group of cells is the inner cells. It's called the embryoblast, which form the embryo and the fetus later on. And the outer cells, it's called the trophoblast, which will penetrate through the endometrium of the uterus. So, as we talked before, the embryoblast, which form the embryo and the fetus, and this outer layer is the trophoblast, which will form later on the placenta and penetrate the uterus. So, the embryoblast at the third week of pregnancy will undergo more differentiation to two types of cell. So the inner cells of the blastocyst will undergo more differentiation to two types of cells: the epiblast and the hypoblast, as we saw here in this diagram. At this stage, the pregnancy sac is called the bilaminar embryonic disc. So this diagram also shows the bilaminar embryonic disc. So if we do a cut section at coronal direction, we will see the amniotic sac and the umbilical vesicle. Between them is the bilaminar embryonic disc, which is formed by two types of cell, an epiblast and the hypoblast. So the epiblast, which in the front of the or in the direction of the amniotic fluid, this is the surface of the epiblast. This is the amniotic cavity, and the epiblast in the direction of the amniotic cavity. This is the cut section or axial view of the amniotic of the epiblast. The epiblast has many landmarks, anatomical landmarks, like the cloacal membrane and the oropharyngeal membrane. The oropharyngeal membrane is formed in the cranial part of the epiblast, and the cloacal membrane in the caudal part of the epiblast. The oropharyngeal membrane will form later on the mouth, and the cloacal membrane will form later on the anus. Just cranial to the cloacal membrane will form a groove at the epiblast, is called the primitive groove or the primitive streak. So as we talk, there is a groove just cranial to the cloacal membrane. It's called the primitive streak. It appears at the midline of the epiblast layer. At the cranial part of this primitive groove or primitive streak, there is what's called primitive node, just dilatation of this groove. It's called primitive node. At the center of this primitive node is the primitive pit. It's important clinically and at the embryological development, as we will talk later on. After the development of the primitive streak and primitive node and primitive bed, there is a process which is very important at the development of the embryo, is called the gastrulation. The gastrulation is the process through which the cells of the epiblast layer break off and migrate toward the primitive bed to form 3 layers. So the cells of the epiblast will differentiate and undergo more division and growth and migrate through the primitive bit, as we told before, at the cranial part of the primitive groove. Then migrate to the underlying layers, as we saw here in the coronal view. It migrates down to the and replace the hypoblast layer to form the endometrium endoderm, and to the space between the epiblast and hypoblast to form the mesoderm, and it replaces the cells of the epiblast to form the ectoderm. So at this stage, the 3 important and the 3 main layers of the embryo is formed at the process of gastrulation. At this stage of gastrulation, after it's finished and mature, the embryonic sac is called the trilaminar embryonic sac or disc. So as we talked, the endoderm is formed by the epiblast cells, which grow and migrate through the primitive bit to replace the hypoblast cells, as we saw down, to form the endoderm. While the mesoderm is formed by the epiblast cells, which also migrate through the primitive bit to the space between the endoderm and the ectoderm. While the ectoderm is the outer layer, is formed by the differentiated cells of the epiblast, which replace the cells of the epiblast, and it remains at its position. So at the end of this process of gastrulation, the bilaminar embryonic disc will be called the trilaminar embryonic disc, as we saw in this both diagram. The left one is called the bilaminar, and the right is called the trilaminar. After that, it's the process of development of the central nervous system. So the 3 layers of the embryo is formed: the endoderm and ectoderm and the mesoderm. And now a process is called neurulation, which the first steps or first process of formation of the central nervous system. The neurulation starts by formation of the notochord. The notochord is a differentiated cells of the mesoderm at the midline, at the mesodermal layer, which is called the notochord. As we saw in this diagram, at the left one is the axial view, and the right side is the coronal view. The coronal view shows this red dot is the notochord at the mesoderm at the midline between the endoderm and the ectoderm. This notochord is very important because it secretes growth factors which stimulate the ectoderm to differentiate the above ectoderm to differentiate to neuroectoderm. And it stimulates also the growth to make this layer over the notochord is thick. So the function of the notochord is to secrete growth factors to make the ectoderm to differentiate and growth more to be more thick and differentiated from ectodermal cells to neuroectodermal cells, and called the neural plate. This neural plate over the notochord start to develop more and more to form a fold, and it will grow up and medially. This neural plate will grow more and make the neural folds, which grow up and medially to form a groove at the central part called the neural groove, and laterally is the neural fold. Between the lateral to the neural folds, a special type of cells is called the neural crest, which is by the green color. So the neural folds move more and more toward each other to fuse finally at the midline. Usually, the fusion started at the middle part of the neural tube. So the plate is grew more, and to make the folds is meet at the midline to make neural tube. This neural tube is formed early or closed early at the midline at the middle part of this tube, and then migrate proximally and distally to close the whole to close the whole neural tube. Also, as we see in this diagram, the green color cells is the neural crest, as we will talk later. So fusion of this neural folds occur at the middle of the neural plate, then cranially at the 25th day, it will close the anterior neurobore, and distally and closed at the 28th day at the distal part, which is called the caudal or the posterior neurobore. So the cranial neurobore is closed earlier than the caudal. During closure of this tube, after it's closed, it contains at its center a cavity which is filled with fluid. This fluid is formed later on the cavities of the CSF in the form of the ventricles and central spinal canal. So the central spinal canal and the ventricles is formed at the cavity, which is formed by the closure of the neural tube. So till this step, we finished the neurulation process. The neurulation process is matured and the neural tube is closed. But if there is any defect at this process, at the previous steps of neurulation, it will form what's called group of congenital anomalies, the neurulation defects anomaly. The neurulation defect anomalies divided to many types according to the site of the defects which is not closed. So, if the whole tube is failed to fuse and closed, it will form what's called cranioreschiasis, which is failure of formation of all the brain and spinal cord at all. And this is not compatible with life and usually stillbirth and death at the embryo life. If the anterior newborn failed to close, it will form what's called the anencephaly, with failure of the formation of the brain and part of the meninges and skull bone and part of the scalp. If the failure of closure at the distal part or the caudal part, it will lead to what's called the spinal dysarthrism or open spina bifida and closed spina bifida. The anencephaly and anencephaly also were parts of the failure of closure of the neural tube, which, as we said, all of these groups of congenital anomaly is called the neurulation defect anomalies. After the neurulation and closure of the neural tube, it usually separated from the overlying ectoderm. As we see here in this diagram, after closure, it's go inside and closed by the overlying ectoderm and the neural crest cells. So, after this process, we will go to another steps or another process of embryological development, which is called the post-neurulation development. The post-neurulation development usually started by the growth of the cranial part of the neural tube and to form three vesicles, which is called the primary brain vesicles. This primary brain vesicles is the borencephalon, mesencephalon, and rhombencephalon, which will form later on the brain, the cerebrum and cerebellum and the brain stem. This primary vesicles will undergo more maturation and differentiation to form the secondary, the five secondary brain vesicles. So the borencephalon, which one of the most cranial part of the primary vesicles, will differentiate to the telencephalon and diencephalon, while the mesencephalon will stay the same as the mesencephalon, will not divide it. The rhombencephalon, which is the most caudal part of the primary brain vesicles, will differentiate to the metencephalon and myelencephalon. So, the five secondary vesicles will form the brain and brainstem and the cerebellum by these five vesicles. As we see also in this diagram, the primary vesicles will differentiate it to the secondary vesicles. Also, this diagram shows a coronal view and lateral view of the primary vesicles, which will differentiate to the secondary vesicles. The primary vesicles, of course, three, which developed at the fourth week of pregnancy. The secondary brain vesicles is differentiated at the fifth week of pregnancy, and will later on make a derivative of the central nervous system, as we will talk later on. So, the derivative of the secondary vesicles, it's according to the secondary brain vesicles. So, the telencephalon will form the cerebral hemispheres, caudate, putamen, amygdala, colostrum, lamina terminalis, and olfactory bulbs and hippocampus. While the diencephalon will form the thalamus, epithalamus, and subthalamus, and the hypothalamus. And the neurohypophysis, pineal gland, globus pallidus, mammillary bodies, and optic chiasm and nerves, retina, epithelium of the iris, and ciliary body. While the derivatives of the mesencephalon is the midbrain. The derivative of the metencephalon will form later on the bones and the cerebellum. The myelencephalon will form the medulla oblongata. And the caudal part of the neural tube after the myelencephalon will form the spinal cord. This table summarizes the primary and secondary vesicles and its derivatives, and also the cranial nerves and ventricle. For example, the telencephalon will contain inside it the lateral ventricle, which is previously the central cavity of the neural tube. The diencephalon will contain within it is the third ventricle. The mesencephalon will contain the cerebral aqueduct. Metencephalon will contain the proximal part of the fourth ventricle, and the myelencephalon will contain the distal part of the myelencephalon. While the neural crest, as we talked before, which surround the neural tube, and as we talked before, is specialized type of the neural tube cells, which separated from it and become surrounding, will form the cranial and spinal and the autonomic ganglion. Also, it contains the it will form later on the bia and arachnoid. Also, it will form later on the Schwann cells, melanocytes, and carotid body. Also, it will form later on the suprarenal medulla and the ganglion cell layer of the retina. After that, the secondary brain vesicles also already developed and start to form the derivatives of the secondary brain structures. The next process is the more maturation of the derivatives and the structures of the central nervous system. It's formed and it's done by the formation of the neural cells and the neurons and its migration. So, the neural tube is lined with neuroepithelial cells. This neural tube is contained inside it the neural canal. This neural canal is lined with cells is called the neuroepithelial cells. As we see here in this diagram, this is the left one of these cells is the neuroepithelial cells. These neuroepithelial cells which surround the neural canal will differentiate to an important type of neural cells and a very important cell in the development of the central nervous system called the radial glial cells. These radial glial cells will differentiate to types of cells, as we will talk later on. Also, this diagram shows the neuroepithelial cells and the its differentiation to the radial glial cells. So the cells with the yellow color is extended from the wall of the ventricle to the outer membrane or the basement membrane of the neural tube, with its processes projected from the wall of the canal to the basement membrane. These radial glial cells will be divided into many types of cells. First, it will copy itself to more growth and multiplicate the number of the cells. So it will form, it will divide to form the more new radial glial cells. Another types of cells is formed by the radial glial cells is the intermediate progenitor, or called neuron progenitor. So these radial glial cells will divide also to one of the important types of the neural maturation is called the intermediate progenitor, or the neuron progenitor. This neuron progenitor or intermediate progenitor, as we see in this diagram, which is called bipolar cells, bipolar neural cells. These bipolar neural cells along the process of the radial glial cells, which we will see here, and it will migrate along the process of the radial glial cells from down to the outside in the direction of the basement membrane. So this is the migration of the neurons from the paraventricular and paraspinal canal to the cortex. So the cells is formed by, and this intermediate progenitor will be differentiated to and specialized to bipolar cells. These bipolar cells, the glial cells, to the basement at the cortex. Also, this diagram shows the neural progenitor at the base of the radial glial cell, and will be specialized and differentiated to the bipolar cells, which is above it at the level of the cells, and then migrate along the process of the radial glial cells in direction of the cortex to reach to the final position of these neurons at the cortex. Also, the radial glial cells, one of its division is the macroglia. Also, these radial glial cells will differentiate to another type of cells. It's called the macroglia. This macroglia is the astrocytes, oligodendrocytes. This cells is the support of the neurons and give the nutrition and maintenance of the blood-brain barrier and myelination. So, to make a revision about this radial glial cells, it divides into three to make more cells of radial glial cells. Also, it divides to the neural progenitor, or called intermediate progenitor, which will form later on the neurons. Also, it's divided to form the macroglia in form of astrocytes and oligodendrocytes. So, after this radial glial cells finish to make multiplication and migration of neurons and make all the macroglia, its function is finished. And the process which extends from the neural tube to the basement membrane outside is regressed. And the cell itself is located and located at the paraventricular area, and make the lining of the ventricle and the central spinal canal in form of the ependymal cells. So this radial glial cells will form finally the ependymal cells at the lining of the ventricle and the central spinal canal. Okay, During this process, the previous process, which we will talk previously, the maturation, the post-neurulation, maturation, and the maturation and the migration. There is some defects occur during this process of the maturation and migration. This group of congenital anomalies is called the post-neurulation defects, which is also known as closed lesion or skin-covered lesion, also known as migration defects. These migration defects can be divided to cranial and spinal. The cranial in the form of microcephaly, hydrancephaly, holoprosencephaly, lysencephaly, schizencephaly, and macroencephaly. Heterotopia and cortical dysplasia also is types of the post-neurulation defects. The spinal post-neurulation defects in form of the split cord malformation and the hydromyelia and syringomyelia. So this one type of the post-neurulation defect is the hydrancephaly, which absence of the supratentorial part of the cerebrum and part of the diencephalon, and replaced by CSF. So I will go rapidly about this because the time is not enough. Also, another type of the post-neurulation defect is the holoprosencephaly, which is failure of the telencephalic vesicles to cleave into two cerebral hemispheres. There are many types of the holoprosencephaly in the form of allobar, semilobar, and lobar. Also, one of the post-neurulation defects is the lysencephaly, which is maldevelopment of the cerebral convolutions, mostly due to arrest cortical development. There are also types of lysencephaly, agyria, brachygyria, and polymicrogyria. Also, another type of the post-neurulation defects is the schizencephaly, which is cleft that communicate with the ventricle, the cleft lined with cortical gray matter. Usually, it's two types, open-lipped or closed-lipped. This is the open-lipped schizencephaly, which communicate between the ventricle and the cortex. Another one is the closed schizencephaly, which opened from the ventricle to the parenchyma. Differentiate from the borencephaly by the lining cells of the cyst or the encephalomalacic part of the brain. So if it's gray matter, that means it's schizencephaly. While if it's white matter, it's mostly due to borencephaly. Another type of the post-neurulation defect is the heterotopia, which is abnormal fossa of non-enhancing gray matter. And from the subcortical white matter to most commonly the subependymal lining of the ventricle. So during migration, as we talked before, during migration of the neurons, there is a defect which arrests the which will arrest the neurons to make it in abnormal position. It results from arrest radial migration, presentation usually seizure. Okay. Another type of the post-neurulation defect is the cortical dysplasia, which is cleft that does not communicate with the ventricles. It's opened from the cortex to some deep of the brain tissue, as we see here in the MRI and in the gross anatomy. Usually is presented with seizures. Also, another type of the post-neurulation defect is the microcephaly. Also, another type is the macroencephaly. The spinal post-neurulation defects in the form of the split cord malformation, which is two types, diastomatomelia or diplomylia, which is type 1 and type 2. In split cord malformation, type 1 is called diastomatomelia, which is two cord in two separated dura, and there is a separate between the two cord is the cartilaginous or bony spur, separating both the spinal cord, while the type 2 is two hemichord in the same dura. This is a type 2 within a single dura tube, separated by non-rigid fibrous median septum. It's associated usually with tethered cord. The management is removal of bony or cartilaginous septum, detethering, reconstruction of dura. Also, another type of the spinal post-neurulation defect is hydromyelia and syringomyelia. Thank you for your attention.
Thank you very much, Dr. Ismail, for this presentation. I think Dr. Mujahid is still here. Dr. Mujahid.
Dr. Ahmed.
Yes. If you have
Finished?
any, I think Dr. Ismail already finished. If you,
Yeah, thank you, Dr. Ismail, for this nice presentation.
you can
And for
stop
the
the screen sharing, Dr. Ismail.
participants, any question, any discussion?
Dr. Ismail, you can stop your screen sharing. I will see in the chat if there is any question or any comments for Dr. Ismail. I don't see here any questions. If anyone has a question or a comment for Dr. Ismail, please go ahead. I think there is no questions. Dr. Mujahid,
no questions. I want to thank Dr. Ahmed Al-Jaradi for this nice preparations and continuous medications, continuous education lectures of the Yemeni Society.
Thank you, Doctor, for your nice work. That's all by your support.
Dr. Aiso and Dr. Ismail for the two nice presentations today. Thank you, Dr. Ahmed.
Thank you, bro. Thank you for all of this.
So I think there is no more questions. So I think we can conclude our third session of the comprehensive neuroanatomy educational course. And thank you, Professor Dr. Aiso Kabaki and Dr. Ismail Mansour for this amazing presentations and this informative lectures. And I think we can conclude today's sessions, and we are waiting for you next Friday, inshallah, with more presentation and more lectures, inshallah. So thank you, everyone.
Inshallah. Thank you, Dr. Ahmed. Thank you, Dr. Ismail. Thank you, Dr. Aiso.
Thank you, bro.
See you next Friday, inshallah.
See you.
Bye.
Thank you, Doctor.